So Tom Grabowski, wow, this guy's pretty amazing. And you know you're amazing when you only get one sheet of a CV. Probably has about a 90-page CV. Tom's a neurologist in the Department of Radiologies. In the Department of Radiology, and he is a true, and you know, people, it's a hackneyed phrase to say a true triple threat, but he's beyond that. He's the director of our UW Alzheimer's Research Center, and he does this multidisciplinary precision medicine approach to Alzheimer's disease. So he runs the ADRC. He also runs one of the best clinical programs we have. This was thought up about 6-7 years ago. The Memory and Brain Wellness Center. Many of us, including myself, are going to bring our parents to that center to take a look at their brains and understand why. No, I'm not talking about you and your father, Jeff. You're smiling at me. That's not what I was talking about, George.
No, I was thinking about what. No,
never. So anyway, and then the coolest thing is the other thing he does, he's the director of the UW Integrated Brain Imaging Center. Now tell me that's not a cool logo. That is a good logo, whoever did that. So Tom, to just sum up, I can go on for 10, 15 minutes about Tom. Tom's a great clinician. He's a scientist. And he's a leader. He runs all these things, and he is a wonderful guy. I mean, anybody who's ever interacted with Tom realizes what a wonderful asset. We recruited him from the University of Iowa, and we are not giving him back. So, Tom, I can go on more, but I'd rather hear your talk. So, thank you very much.
Thank you, Rich. That was a most generous and undeserved introduction. So I'm happy to be here this morning as your guest and to talk to you about Alzheimer's disease primarily. And what I'll talk about is fairly broad, and we'll touch on the themes both of our Memory and Brain Wellness Center and our work in the Alzheimer's Disease Research Center, because I think it's important to show how these goals are synergistic and interactive. Let's see, this does not do this, okay. So I just want to acknowledge our funding from the National Institute on Aging and the Ellison Foundation. I don't really need to say much about this. Alzheimer's disease is clearly one of the biomedical challenges, if not the biomedical challenge of our time. The bullets actually understate this for reasons I'll get to, but about 9% of Americans over the age of 65 have Alzheimer's disease, and this is really the only leading cause of death in the country in which we have made no inroads on the natural history to date. The numbers are going to double and triple in the next decades as the population ages and as we continue to make progress on the other leading causes of disease. And the medical and unpaid caregiver costs, you hear numbers bandied about that amount to something like $1 trillion over the coming years. So it's a lot of impact. So what I want to do primarily this morning is challenge some of the ideas you may have, and clearly the public does have, about Alzheimer's disease. And specifically, we have Alzheimer's disease in a pretty tight frame in the way we think about it. You know, pretty much Alzheimer's disease and dementia are synonymous in the minds of most people. And it really is framed by a sort of catastrophic stereotype. Loss, total loss, everybody in the same boat. But what I would like to point out is the frame is actually much bigger than that. Dementia and loss are really just one part of the picture. And in this frame is a much broader and longer lasting disease than that frame would suggest to you. We talk about mild cognitive impairment. I'll talk about that at length. Earlier stage where there may be subjective feeling of memory decline, but no objective evidence of it. Even we'll talk about the concept of normal cognition with Alzheimer's disease nevertheless. And there are important factors to think about. There's something called cognitive reserve. There's the possibility of engaging retained strengths. There's the possibility of even living well with memory loss. And we're starting to talk seriously about prevention of Alzheimer's dementia. So I'd like to touch on a number of these themes today and help you think about Alzheimer's disease as a broader concept. So we'll talk about these four things: preclinical and prodromal Alzheimer's disease, resilience to Alzheimer's disease, and an evidence for the modifiability of the expression, at least, of the disease. The heterogeneity of mechanisms driving Alzheimer's diseases, if you will. And then I'll spend a little time with this concept of topographic heterogeneity in Alzheimer's disease, because investigation of this, or at least using this as a level of description, is one of the themes of our Alzheimer's Research Center going forward. So with that sort of overture, we have to start with plaques and tangles, which I don't. Is there anybody in the audience that doesn't know about plaques and tangles these days?
I do have one question.
Yeah.
Can you give us the update of how many patients diagnosed with Alzheimer's do not have? There's more data, but there are some that do not have that. Correct.
Well, I don't know if I will agree with that. So pretty much the presence of plaques and tangles is what you need to define Alzheimer's disease. There are patients who have progressive degeneration with syndrome, more or less indistinguishable, that may not have amyloid, so-called SNAP, suspected non-Alzheimer pathology. It's an area of active work. No, no, yeah. So Rich mentioned that I was at Iowa before this, and when I was at Iowa, I had this, as a neurologist, I had this epiphany when I saw this diagram that was in one of Steve Arnold's papers. This was about 1990. And it was also done with Gary Van Hoosen. George, you know Gary, don't you? So this is just a cross-sectional representation of the density of neurofibrillary tangles in Alzheimer's disease. And as a neurologist, and I dare say as neurosurgeons, of course, you recognize a lot of structure in that image, right? This is not disease that equally impacts all parts of the brain. This is disease which is heavily impacting the medial temporal lobe, heavily impacting the temporal neocortex, relatively sparing primary visual, primary sensory and motor, primary auditory regions, and pretty much singling out limbic and high order association regions. That's interesting. That's something that really does explain the clinical phenotype of what I'll call modal Alzheimer's disease. Initially, an initial more or less pure amnesia, leading to progressive loss of higher function and an almost complete sparing, at least for many years, of everything to do with motor functioning and gait. Okay. So Alzheimer's disease is not a truly globally diffuse process. Modern PET-based biomarkers with amyloid and tau tracers show similar structure. Okay, so Alzheimer's disease, first of all, is not something in which the impact is felt evenly by every part of the brain. And it turns out that this neurofibrillary pathology unfolds temporally in this same sort of structured way. So this is formalized in what are called Brock stages. And moving from left to right, you have a more or less medial temporal stage, so-called transentorhinal stages of Alzheimer's disease, limbic stages 3 and 4, and finally isocortical stages 5 and 6. And these correspond more or less to phases of symptomatology of Alzheimer's disease. So you have Alzheimer's dementia corresponding to neocortical investment of neurofibrillary pathology. And as we've all heard, from diagnosis of Alzheimer's dementia until someone may pass away, on average, that's something like 8 or 10 years. But before the phase of dementia, there is an increasingly well-described phase of mild cognitive impairment. And this is a generically, or a deceptively generic term, mild cognitive impairment. It really means that you have measurable episodic amnesia, but relatively intact activities of daily living. Okay, so no impact on living, but a measurable impact on memory. And if you have that amnestic form of mild cognitive impairment and you're 65 years old, the chance that you will move on to mild dementia is about 10% per year, and ultimately probably about 3/4 of individuals will convert from a mild cognitively impaired stage to dementia. So we increasingly think of amnestic MCI as mainly, but not entirely, mainly prodromal Alzheimer's disease. And before that, there is a truly asymptomatic stage. As we'll talk about, this lasts some 15 years or more. So think about that. Alzheimer's disease, fully framed, is a decades-long process, most of which is spent in the pre-demential phases. Okay, and there's a long window, therefore, for an intervention on the natural history, which might delay progression and ultimately reduce or prevent the emergence of dementia. Turns out that this sort of pre-symptomatic Alzheimer's pathology is very common. So these are data that are taken from the, what was Group Health ACT study, now it's the Kaiser Permanente Washington ACT study that Eric Larson and Paul Crane direct. And in serial autopsies of individuals, so does everyone know about this study? So this is one of the leading studies of the natural history of, or the risk of dementia in aging individuals. And it's a random sample of Kaiser's clientele in Seattle, 2,000 subjects at any one time, followed to the endpoint of developing dementia. And there's a big autopsy follow-up program connected to this. So there's a lot of data from the ACT study about what typical aging brains look like at post. And what I'm showing here are basically data from 112 persons who died without dementia. And each bar in this graph is one person. And what you're seeing in blue is the amount of Alzheimer's pathology, green, the amount of Lewy body pathology, and red, the microvascular injury detected in these brains, stacked from the least affected on the left to the most affected on the right. The main point I want to take away is hardly anybody's unaffected. Okay? The great majority of individuals have some degree of low-grade Alzheimer plaques or tangles, even though there's not dementia present. We now can begin to look at the prevalence of some of these factors in vivo. I mentioned amyloid PET scans a little bit ago. I'm showing you across the top 4 amyloid PET scans that have been performed in our center over the last year and a half as part of the IDEAS trial. And on the left are 2 negative amyloid scans, on the right are 2 positive amyloid scans, and you can tell a negative from a positive scan by whether or not you can see the gray-white matter structure in the image, because the non-specific binding, there's non-specific binding in the white matter, but there should not be any binding in the gray matter. That's a negative scan. And you can see the sort of ground glass appearance of the cortical areas in these 2 positive scans. And if you look at the rate of a positive amyloid scan as a function of age, which is the blue curve, and the rate of incidence of Alzheimer's disease dementia, the red curve, you see the more or less parallel curves that are shifted 15 years relative to each other. This is the basis for the statement that Alzheimerizations probably happening about 15 years before emergence of symptoms. And if you look at age 70, the estimated rate of positivity is about 25%. So at age 70, the rate of Alzheimer's dementia is somewhere in the single digits %, but fully 10 times that has a positive amyloid scan. So this is something which is affecting a serious amount of the population. So
I'm sorry, Tom, but amyloid is higher in the dementia. That graph is.
I'm sorry.
I'm just confused by the graph.
So the blue graph is the rate of a scan being positive. The red line is the rate of dementia. Okay. So basically, the scan positivity leads dementia by 15 years. Does that make sense? Yeah. Okay. So,
Are you going to talk about just that?
yeah. I mean, the interesting, an amyloid scan is not a complicated thing. It's up or down. Okay. It's an interesting thing about amyloid. Actually, it's a good point. By the time one has any symptoms, the amyloid scan is not, it's not subtle. The amyloid scan is positive, or the amyloid scan is negative. Yeah, there's very little, ultimately very little regional information in this amyloid scan. No matter whether your presentation is amnesic or visual or language, that amyloid scan will look basically the same. It's not the story, not the same story with the tau tracer. Okay, so what's coming for research purposes? What's coming for research purposes is a fully biomarker-based definition of Alzheimer's disease. This is something which has been trotted out at the last couple annual Alzheimer meetings, and you'll probably see promulgated in the research literature this year. And basically making use of 3 kinds of biomarkers, which are called A, T, and N, A for amyloid, T for tau, N for neural degeneration. And you can get the amyloid marker from spinal fluid A beta, spinal fluid A beta 40 to 42 ratio, or amyloid PET. Similarly, the tau marker from spinal fluid or PET, and the neural degeneration biomarker might be CSF total tau, might be FDG PET, might be cortical atrophy from MR. But if you have a positive amyloid marker, you have Alzheimer's disease pathophysiology. And if you have a positive amyloid and a positive tau marker, you have Alzheimer's disease, full stop. Doesn't matter what your cognition is. Okay. So really, this is a radical separation of etiologic diagnosis from cognitive diagnosis. And we're talking in terms of individuals with a positive amyloid scan, even though they're asymptomatic, having either preclinical Alzheimer's disease or at least preclinical Alzheimer's pathophysiology. Does that make sense? Okay. So if there's a tsunami of Alzheimer's disease coming, I like to say there's a tsunami with a big iceberg in it. People are only thinking about the symptomatic dementia component of that, but the number of individuals that's actually affected in a preclinical way is much larger. Okay. So to sum up so far, Alzheimer's dementia is just the last phase of a decades-long process, and there's a long latent disease period, presumably therapeutic window for the future. It's really not possible to altogether separate cognitive aging and preclinical Alzheimer's disease unless you go and get an amyloid scan. And a large proportion of the aging population has preclinical AD. So I like to think about things this way, separating the idea of degeneration resulting in these physical abnormalities in the brain from circuit disruption or physiologic disruption, which results in the cognitive manifestations. And as I mentioned earlier, there's a radical separation of the diagnosis of syndrome and the diagnosis of etiology, which relies on biomarkers. And if you keep that straight, you avoid a world of hurt in this field. But you can see, as I started the whole lecture, most people think immediately of dementia when they think of Alzheimer's disease. So there's a number of factors which conspire to cause this degeneration. Amyloid, Alzheimer-specific pathways being just one of them. And there are factors that conspire to slow the degeneration or the expression of symptoms. And we'll talk a lot about this phenomenon of cognitive reserve in a minute. And then there are some other short-term factors that impinge here and are a focus of medical management. Everybody with me so far? Can you hear me okay? Okay. So the next thing I want to point out is, we're talking about Alzheimer's disease, but comorbidity really is the rule, at least for aging and aged sporadic late onset disease, let's say. That's the same sort of data from the Kaiser Act study, but these are autopsies of patients with dementia, so all the colors are shifted upward. But the point is still that it's rare to have only blue. Usually there's a component of microvascular injury and often a Lewy body component as well. And statistically, Alzheimer's disease explains about 45% of dementia and microvascular injury, about 33%. So in the real life, this nice mapping of cognitive onto pathological stage doesn't really happen that way. It sort of slants, and you can kind of be dragged back by this comorbidity. So you may have mild cognitive impairment at an earlier Braak stage. You might have dementia at a Braak stage, which should normally be associated with mild cognitive impairment, for example. The good news is you can also lean into this disease. You can also push these relationships to the right, such that you might only have mild cognitive impairment with BRCA5. We also see patients who come to autopsy who have BRCA6 and didn't have any record of cognitive impairment in life. And, so what explains that? So this is the phenomenon of cognitive reserve. And this has a lot to do with your lifestyle. It has a lot to do with your habits over midlife. One study that I'll point you at was done at the Mayo Clinic, published in JAMA Neurology about 3 years ago now. This is the other leading natural history study of aging and dementia risk in the country besides the ACT study. There were a bunch of studies funded back in the 80s. These are the two surviving continuously funded studies. So they studied basically Olmsted County, looked at things like years of education, how cognitively challenging people's employment has been, and what are called mid and late life cognitive habits. And this is just divided pretty simply into low and high cognitive habits. Low if you had less than 3 hours a week of cognitively demanding activity, and high if you had at least 3 hours. Okay. You went to the mental gym 3 times a week, if you will. They need a better graphical artist for how they represent their data. But I thought I'd show this anyway. So what you have are on the y-axis, this is a kind of an amalgamation of the education and occupation scoring. And then the lines indicate the difference between low and high cognitive habits for the 3 groups of education and occupation, low, medium, high. Basically, what I wanted to show is you have the same sort of leaning in relationship that I showed a little while ago. So the difference between a low education and occupation score and a high one is somewhere around 4 or 5 years difference, in which, well, I should back up. So they modeled the effect of these factors and found that you had 2 factors that were important: your education and occupational attainment and your cognitive habits across mid and late life. And they're expressing these results as if you're modeling an 80-year-old person currently cognitively unimpaired carrying an E4 allele. Okay, so an 80-year-old person carrying an E4 allele is someone who's at high risk for developing Alzheimer's dementia. Okay, for that sort of individual, the time to develop dementia is going to be, on average, this many years, 4 or 5 years if they have a low education and occupational attainment, and 8 or 10 years if they're in the high category. Similarly, there's a 3 or 4 year effect of their cognitive habits, and these factors add together. In other words, you can push back by up to a decade the time at which your Alzheimer's disease will become symptomatic for you by your midlife habits. Okay, that's the phenomenon of there it is. That's the phenomenon of cognitive reserve. You want to be up in this quadrant. So here's a fairly direct demonstration of this concept using modern tau PET imaging. So here we're looking at high educated and low educated patients diagnosed with Alzheimer's disease having equivalent degrees of cognitive impairment. So if you look high and low educated versus controls, and this is what's colored on the brain images here is tau tracer. So you see a lot of tau pathology in the high and the low groups versus the control. But if you compare the high versus low education groups who have equivalent amounts of cognitive impairment, equivalent amounts of dementia, it takes a much larger tau load to drag the high educated group into that zone. And if you look at how this breaks down by Braak stage, it really shows up in the higher Braak stages, 4, 5, and 6, where the high educated groups have very high degree of pathology to bring them to the same degree of cognitive impairment that a low educated group will have with much less tau pathology. The other point I wanted to make is if you just do the thought experiment now, where let's suppose one of these current anti-amyloid trials turns positive, right? And we find a way to delay the natural history of this disease by 5 years. Okay, so now mild cognitive impairment won't be 5 years long, it'll be a 10-year long phase, let's say, in the disease process. You think about that compared to the current 5-year phase, look at these effects of lifestyle. You project them onto the x-axis. These effects get much longer with a delaying agent. So my point is that these lifestyle interventions are here to stay. There's something that is going to potentiate any treatment that we develop that alters the natural history of this disease. Okay, so then reframing Alzheimer's disease intervention. So early diagnosis and intervention is a strategy to prevent dementia, and lifestyle interventions are an important component. The evidence is really good that lifestyle affects your risk. The evidence is not so strong that you can make interventions on the lifestyle once you have symptoms and change your risk. There is some evidence, but if you hold it to a standard of randomized clinical trials and what will the National Academy of Sciences stand behind, that's a little problematic yet. But it's pretty clear to me that cognitive reserve and vascular health are likely going to enhance the benefits of emerging medical treatments. And then the other thing I wanted to say about the clinical side of this is the same sort of insight, that all parts of the brain are not equally affected by this disease, is the scientific basis for approaching this with a strength-based kind of paradigm. In other words, because Alzheimer's disease doesn't affect all the areas of the brain, there are going to be areas which are relatively better off and are resources for patients and provide a basis for compensation. So the best established example of this is that Alzheimer's disease more or less spares basal ganglia and cerebellar regions, at least in up into the severe stages of the disease. And these are the systems which are important for procedural or habit learning. So there's an entire therapeutic program which the Mayo Clinic has arranged around this concept called HABIT. I love the acronym, Healthy Action to Benefit Independence and Thinking. This is Glenn Smith, who was at the Mayo when he developed this. Basically a 50-hour, 2-week boot camp for patients with amnestic mild cognitive impairment. And one of the centerpieces is this calendar habit training. It's a very simple concept. You always write everything on the same calendar in the same pocket the same way. Did I say the same way every time? and you go through this with a partner, and it's sort of drilled into people. But Alzheimer patients can acquire habits just as easily as normal individuals can, and you get a useful habit which can prolong your independence, right? So that's one example, but I think this idea of compensation using spirit systems is just beginning to take hold. All right. The creative impulse is another relatively spared faculty. I won't spend much time on this, but this is artwork produced by a patient with Alzheimer's disease. We have a very active community in Seattle interested in arts engagement in patients with Alzheimer's disease, including programs at the Fry Art Museum in our neighborhood here. In fact, I didn't realize this, but museums don't just sit there and wait for clientele to come visit their museum. They actively cultivate sectors of the community to take advantage of their programming. And most museums will reach out to children. The Fry Art Museum here on First Hill is reaching out to seniors and has this very active creative aging program. And we're strongly considering partnering with them for some of our therapeutic programming going forward. You can imagine a very non-stigmatizing environment to do that. So, clinically, in 2017, if you come to our Memory and Brain Wellness Center, besides accurate and early diagnosis and improving symptoms with medications like cholinesterase inhibitors, we're also about helping people build brain health to maintain their cognitive reserves, reframing living with memory loss with a more positive vision, this concept of therapeutic strengths-based engagement. And of course, we systematically invite them to research. And if you want to know more about us, you should look at our webpage. So with that, I'm going to kind of shift to the second part of the talk, which is more scientifically focused. And I'm going to begin with this diagram, some version of which has been drilled into everybody who comes to any Alzheimer meeting for the last 4 or 5 years. And I'll call this the canonical sequence of Alzheimer's disease biomarkers. And on the x-axis, you have basically disease stage or time with disease from this latent or pre-symptomatic epoch through amnestic mild cognitive impairment to dementia. And on the y-axis, how often a given biomarker is abnormal. And what you see is spinal fluid markers of amyloid beta, markers of amyloid deposition, which you can get from spinal fluid or PET scans, turn positive first, then measures of tau, later measures of neural degeneration, and finally cognitive measures. Okay. There's the confounding issue of sensitivity of these different techniques, but this is roughly in the order that you would expect if the amyloid cascade hypothesis were true. So the amyloid cascade hypothesis has been the leading paradigm in understanding Alzheimer's disease pathophysiology for the last 20 years or so. Pretty much have to start here. The idea is that deposition of amyloid beta in some way is the initiating insult for Alzheimer's disease, and there's some toxic effect of A beta on the neural environment, probably as oligomers, not as plaque, and that neurodegeneration is a downstream effect. There's been a lot of work done with this. It's very complex. Just to show you one example, there's evidence that amyloid oligomers can bind with the help of the prion protein to this metabotropic glutamate receptor, cause signaling to cause kinases like fin kinase to phosphorylate tau and phosphorylate NMDA receptor components, and allow dysfunction of the NMDA receptor such that you have a hyperexcitable cortical membrane. That's one mechanism of amyloid tau interaction. been popularized recently because there is increasing evidence for hyperexcitability of hippocampal structures, especially in Alzheimer's disease. And this is a hot area of current work. But as I mentioned, it's more complicated than that. It's not just hyperexcitability and synaptic dysfunction, but there's evidence of microglial activation. There's evidence of a primary vascular effect. And so all these pathological factors are potentially playing a role in this amyloid cascade, converging on effects that promote cortical neuronal loss. This amyloid cascade has come under a lot of attack because of its failure to sort of predict what sorts of intervention which would be therapeutic in Alzheimer's disease. You hear of a lot of failed clinical trials, a lot of agents which have come out of mouse models and have been ineffective in human trials. But I would like to just point out that you really only need to know one thing to know that amyloid is toxic and relevant. And it's just simply, if you have an extra copy of the amyloid precursor protein gene, you get Alzheimer's disease early. If you're one of those rare genetic cases in which there's a duplication, that's enough to cause Alzheimer's disease. This is from a large British series published in 2012. You see they had 5 probands that were detected in this series. These are the black bars are where their duplications are. Here's the ATP locus on chromosome 21. And they had, autosomal dominant preclinical, early onset Alzheimer's disease. Interestingly, many of them with seizures. That's just one of the sort of Mendelian forms of Alzheimer's disease. These early onset cases are all cases in which there's a dysfunction in the amyloid processing, either production or processing pathways. The presenilin 1 mutations are the dominant ones in terms of numbers. Presenilin 2 was discovered here at the University of Washington by Tom Bird, Jerry Schellenberg, and colleagues. Interestingly, has seizures as a frequent accompaniment there as well. As we'll come to in a minute, it can't be the whole story, okay? It's just, this is part of the story for sure. So Rich mentioned earlier this concept of precision medicine. So precision medicine is something which is all the rage now. When we wrote our Alzheimer's disease competing renewal 3 or 4 years ago, was before Barack Obama's State of the Union address, in which precision medicine became a national mission. So we've described our center's mission in these terms, but we're sort of being forced to be really clear what we mean by that at this point, because precision medicine has become as much a buzzword as translational medicine was before that. What we mean is, first of all, acknowledging that there is a heterogeneity of mechanism in Alzheimer's disease, and that the particular molecular driver in any one individual may vary, okay? So we wouldn't want to stratify by disease mechanism or genetic risk, then detect the disease early, ideally while still latent, and align treatment with the molecular driver of disease. This third bullet is aspirational at this point in the field, but it's an important concept to test. The best test of this currently happening is in the realm of anti-amyloid therapy for groups in which we know that amyloid is the driver. These anti-amyloid approaches range from, believe it or not, vaccines. This is something which was, the field was very briefly extremely excited about 15 years ago, based on mouse model work. There's a theme here. And this didn't work out well in human trials because the vaccine caused a vasculitis in a number of the patients, sort of amyloid angiopathy-centered vasculitic picture. And the field dropped back to a passive immunization approach using monoclonal antibodies like these ones. And now there are beta secretase inhibitors available. There are a number of what I would consider truly precision medicine trials that are ongoing. One of them is the DIAN, or Dominantly Inherited Alzheimer Network Trials Unit, which is basically studying families that have presenilin 1, 2, and amyloid precursor mutations in them, in individuals who are currently asymptomatic but carry the mutation. We are a site for this study that Sumi Jadav leads that. There's also an Alzheimer's prevention initiative that's taking place largely in South America, where there's a very large kindred with the presenilin 1 mutation. This is using this monoclonal antibody. And then interestingly, there's work emerging in Down syndrome, because remember, Down syndrome is trisomy 21. So all the Down patients have an extra dose of amyloid precursor protein. And 80% of Down's patients develop Alzheimer's disease, and usually prematurely. So there's actually work on a vaccine for Down syndrome going on. The jury is out on all of this work, but this is really the acid test. I'd say the acid test of the amyloid hypothesis, and whether or not we're going to be able to modify it with these immune approaches. We do know that these antibodies do remove amyloid burden from the brain. So this is work published in Nature, I guess it's 2 years ago now, showing from baseline to 1 year, no change in amyloid burden on an amyloid PET scan with a placebo, but a dose-related reduction in amyloid using a monoclonal antibody called aducanumab. So we can pull it out. Is it too late to do so, is the question. And I thought I'd mention, although it's not really a precision medicine trial, that there are trials ongoing now for intervention with anti-amyloid agents in asymptomatic individuals with positive amyloid scans. You ask, how would somebody know they have a positive amyloid scan? It's because they volunteer for the research study. So if you are someone who is 65 years or older and have normal cognition, that's your only qualification. You volunteer, have a PET scan. If you're positive, you can be randomized to receive an oral beta secretase inhibitor or not. And that's a trial that we're a site for. And if you're interested, you can talk to me. Okay, but I said we're not going to say amyloid is the only driver. If you now look at genome-wide association studies that have been done on Alzheimer's disease for sporadic late onset disease, you see a panoply of genes which are implicated, and genes and proteins which are implicated. And it's interesting that none of them are, or very few of them, have to do with amyloid processing. You have, so this is a diagram that was published by Alison Dode's group. On the x-axis, you have how frequently you see these particular alleles in the population. And on the y-axis, how much risk those alleles confer. We talked about these rare, high-risk alleles a little while ago that cause Mendelian early onset disease. But you have this big group of common variants that affect your risk, 1%, 2%, something like that, but conspire to tip your vulnerability and have to do a lot with immune responses, especially innate immune signaling, and also with endosomal function, for example. And it's pretty clear that late onset sporadic disease may have a different driver. If you look at the early onset sporadic AD cases, they also turn out to be enriched in these same risk alleles. So to make an enriched sample for study, looking at sporadic early onset disease appears to be a good idea. There's work going on in a number of these pathways in our center. Many of you probably don't yet know Jessica Young, who joined pathology and the Institute for Stem Cell and Regenerative Medicine last year. But she is working on endosomal pathways using this really exciting new approach of bypassing the mouse model and working in human cell models that are generated from induced pluripotent stem cells. We can now generate these stem cells from meninges at autopsy or from skin biopsies on living individuals, and you can start to isolate risk alleles, and then using CRISPR-Cas9, you can actually experimentally study these risk alleles in the dish. I won't go further into it right now, but just suffice to say that's a pretty exciting direction for the work right now. Across the center, there's a number of projects that are going on, working on how basic cellular aging pathways factor in. That's Matt Kaberlein's work. How different forms of A beta might be tagged or interfered with. That's David Baker's work in the Institute for Protein Design, neural inflammation, endosomal trafficking, and so forth. Okay, so now I want to turn back to the biomarker images. Remember, I said we have amyloid scans. Amyloid scans detect amyloid plaques. These have a pretty stereotypical distribution. They lead symptoms by 15 years. They don't change over time. We use them to certify diagnosis, to qualify people for clinical trials. Under the IDEAS trial, we were able to use this clinically and get it paid for by Medicare for a while to tell whether Alzheimer's is playing a role in a tough case. And in contrast, we have tau scans now, tau scans which will come online for us at UW next month. These detect the protein that forms neurofibrillary tangles. Tau scans demonstrate much more variable impact of the disease that correlates quite well with symptoms. You can really use this to delineate disease impact, and I think in the future to track treatment. And that's caused us to take a closer look at plaques and tangles. And when you look at it, although both amyloid and tangles accumulate in the brain, they don't always coincide. For example, amyloid accumulates really early in the basal frontal lobe and in the basal occipital lobe, but that's not where tangles happen first. It's not where tangles happen hardly ever in the occipital region. Conversely, tau accumulates really early in the basal temporal lobe, but this is in the place where amyloid tends to occur early. Other areas like the medial parietal lobe show both markers. So that's curious and suggests that these markers aren't marking the same thing, right? And that the process is playing out differently in different parts of the brain. We occasionally run into outlier patients that are exceptions that prove the rule. How many people have seen the Ishihara plates? This is a traditional color blindness test. This is a control plate that everybody can read. That's experimental plate. We run into patients who are just unable to read these. Okay, they present with visual dysfunction. And they go to their optometrist and then their ophthalmologist, and they ultimately wend their way to our clinic, and they turn out to have posterior cortical degeneration with low metabolism in the parietal and occipital region, and this proves almost always to be a species of Alzheimer's disease. And the so-called posterior cortical atrophy or visual variant of AD. And if you look at this, it's really the exception that proves the rule. Here's on the lower half, the visual variant, on the upper half, the modal amnestic variant, and here are the tau scans on the right. In amnestic AD, medial temporal and basal temporal tau, and posterior cortical atrophy, relative sparing of that area, with heavy investment in parietal occipital lobes. And so we have this phenomenon of topographic subtypes of Alzheimer's disease. For those of us who work in the memory clinic, this is not surprising. We've seen this and recognized this for a number of years, but thought they're kind of freak cases, and maybe we tend to see them in a tertiary center. And we would see all kinds of subtypes, like besides posterior cortical atrophy, there's a progressive aphasia variant. There's a cortical basal syndrome due to Alzheimer's disease. There's an executive predominant Alzheimer's disease. Here's a good kind of demonstration of that. Here's two patients on the left and on the right that we've seen in our memory clinic in the last couple months. Here's one, an amnestic presentation, a lot of hippocampal and anterior temporal atrophy, but not much medial parietal atrophy. On the right, another patient, same presentation, amnestic Alzheimer's disease, relatively spared hippocampal and medial temporal structures, marked medial parietal atrophy. So you have a hippocampal, limbic predominant and a hippocampal sparing sort of species, both with a clinical syndrome that is similar. So we've gotten very interested in this sort of topographic and cognitive variability of Alzheimer's disease. In the ACT study and across 3 or 4 other studies which have now been studied by Paul Crane and his group, it turns out that these variant presentations, say disproportionate language impairment or disproportionate executive impairment, occurs in something like 40, 45, approaching 50% of sporadic late onset cases. Okay, so this is a common sort of variability, not something that's just a phenomenon in a specialty clinic. What does that mean? So one thing that we're interested in is whether these reflect different genetic and mechanistic influences. Paul's group and groups before him have shown that the limbic predominant or disproportionately amnestic presentation is associated with ApoE4, for example, but preliminary indications from Paul's aggregation of the public data sets that I described a minute ago suggest there's additional genetic heterogeneity, and that work is under review right now. So, we have cognitive variants and topographic variants that are closely related. It's commonplace, actually. There's preliminary evidence that genetic background varies across these cognitive subtypes. And then this point that I made a little while ago that amyloid and tau disease markers have different expression and conjunction over the brain space. So we're wondering, are these findings trying to tell us that different mechanisms are playing out even within individuals? Or not put another way, if you think about Alzheimer's disease in this kind of framework where you have a background of genetic factors, environment, and comorbidity, some sort of insult going on, maybe resilience mechanisms at play, but the net result is neurodegeneration. Again, filtered through these resilience mechanisms underlying cognitive reserve, which have something to do with plasticity or systems in the brain, resulting in a clinical phenotype. But I've just said that clinical phenotype is quite heterogeneous. So the question is how much that reflects mechanistic heterogeneity. And what our insight is that topographic heterogeneity may be one level at which to analyze this relationship. And we're actually starting to think about phenotype as a topographic phenotype, if you will. So phenotype is no longer a label, but phenotype is something that's starting to look a little image-like. It's the expression of biomarker over the brain space. And the general hypothesis is that the topographic expression of the disease is related to disease and resilience mechanisms. And we're kind of pursuing that in 3 different ways. Like Paul is sort of in silico using analysis of open data sets for genetic discovery and hypothesis generation about what pathways may be implicated. In vivo work that's being done by the clinical arm of the ADRC, Sumi Jadev, Jessica Young, and others, in which we ascertain these variants with neuropsychology and tau imaging and use induced pluripotent stem cell models to build in vitro models. And then postmortem work, which we'll call spatially extended omics. And this is a really exciting recent development where we're teaming with the Allen Brain Institute to apply some of the brain initiative tools that are being used for the human cell atlas to Alzheimer's disease. Okay, so almost done here. So Alzheimer's reframed and spatially extended then. So there are these natural topographic correspondences across pathological imaging and clinical data. And thinking about a spatially extended phenotype is just a natural extension of current efforts to research define Alzheimer's disease using biomarkers. So the tau marker is going to be especially important here, but there are others that are coming. There's a synaptic integrity PET tracer, which is coming, for example. This sort of paradigm supports much richer data analysis, potentially much more sensitive to detect concurrent disease processes using spatial and multivariate techniques. And by the way, makes contact with knowledge of brain organization and software from the neuroimaging community. So that's a satisfying connection for me. And then finally, biological resilience can be approached in the same framework. Okay, so to sum up, I think there's some very, obviously I think about this like a neurologist, right? There's themes of localization here. There's themes of trying to understand what the anatomical aspects of the disease are trying to tell us about mechanism. So there are important insights to be gained from the topography of AD in the brain. It explains the cognitive profile, the tau and neurofibrillary pathology distribution in particular explains cognitive profile. It helps us detect and operationalize resilience. It forms the scientific basis for these strength-based treatment approaches, provides clues to mechanistic heterogeneity, and in general, I think is a very useful level of description to support deeper research work. And then by reframing Alzheimer's disease to include all its stages and the diversity of its expression across individuals, our clinical and research efforts can become more synergistic. Helps us to reduce stigma and encourage early diagnosis. If we don't do that, our subjects are not going to come to us. And then helps us advance a vision of precision medicine for Alzheimer's disease. So I've mentioned the work of a number of my colleagues today, so I thought I would acknowledge them here. And thank you for your attention and take any questions. Thank you.